All right. Hey, everybody. We are going to just in case we weren't live 4 seconds ago, we're definitely live now. And I am so excited that we're finally to our first session. We are picking up speed and solving our technical challenges today. So with me, I have one of our amazing faculty, Dr. Divya Chander. She's a physician and neuroscientist who trained at Harvard, UCSD, UCSF, and the Salk Institute. She's been on the anesthesiology faculty at Stanford University since 2008 and neuromedicine faculty at Singularity University since 2010. Her postdoctoral training in optogenic technology was conducted in the laboratories of Carl Deisseroth and Louis de Lesia. I'm sorry if I'm butchering your names, at Stanford, where she used light-activated ion channels inserted into DNA to study sleep and consciousness switches in the brain. In the operating room, which is where she's been spending a lot of her time lately, she applies EEG technology to understand what human brains look like when they lose and regain consciousness, and has recently developed a precision medicine initiative aimed at understanding genetic variability in responses to anesthetic drugs. And so with that, Divya is going to be with us here today to kind of give us the basics of viruses, coronavirus in particular, and COVID-19. And with that, I'll turn it over to you, Divya. Thanks for being here. And it looks like we might have a slight delay with Divya since the session went live, but she will hear this eventually and we'll go live.
Hi, good morning, everybody. It's my pleasure to be here with you and to open our first virtual summit on COVID-19. One of the things I wanted to do was set this up for you because there is so much that is unknown about this particular virus. And one of the very first things that is unknown is what exactly is a virus. So let's start at the very, very beginning. What you see here is a reconstruction of what this virus looks like. And all of those red things that you see sticking out that look kind of like triangles, they are spikes. They sit on the surface of this particular virus. What a virus is simply a nucleic acid. It's DNA or RNA. And it's got a protein coating around it. Sometimes it's got a fatty coating around it, but it can't make more of itself. It's not like a normal life form. It can't reproduce. And so the only way it can reproduce is by infecting other cells. So let's take a look at what that looks like here. So here's our virus, and it has those spiky proteins on the outside. And this is a very recent reconstruction of what those proteins actually look like. And why is that important? Those are the proteins that actually bind to receptors on the surface of human cells. And you can see this in the picture. I'm not sure if you can see my mouse in this particular diagram, but here's the actual spike protein of the virus. And there's this sort of red center. And then you see this sort of greenish thing underneath with these purple things that are sticking up. That's the human cell that is binding to the COVID spike protein. So why is this important? Well, that's how the coronavirus gets in. That's how it gets into something like your lung tissue. And you can see that once that receptor binds to the spike protein, it's our proteases. We have enzymes within our cells that can actually break apart the spike protein and activate it. And once that happens, it gets into the cell. Once it gets into the cell, it can make more copies of itself. So then what it does is it hijacks the machinery that our human cells use to normally make our own proteins. So it injects its RNA, it's an RNA virus, it gets turned back into DNA, and then it gets sent through the entire system to make more copies of itself. It gets turned into messenger RNA, and then it gets turned to protein. And those viral proteins get reassembled, and then they get sort of secreted off out into the world, and they destroy the cell that was making them. And that's often how we get sick, because that's another major question. How does the COVID virus make us sick? It makes us sick because in order for it to reproduce, it has to destroy our cells. And the way in which it targets our cells is it looks for proteins on the surface of our cells, and they're mostly present in our lungs. And that's why the symptoms of getting sick with COVID virus look so much like the common cold, if it's a mild case, or a really bad flu. So if you begin to feel things in your nasal passages or in the back of your throat, could be a normal cold, but it could be the coronavirus beginning to take hold. And then it begins to move deeper and deeper into your lungs. And it's usually people who are elderly, people who have pre-existing conditions, that it begins to move deeper into their lungs. And what eventually happens is that it destroys the air sacs that your lungs use to communicate with the blood. And it's harder and harder for them to exchange oxygen and carbon dioxide. And that's why people who have really bad cases of coronavirus find it hard to breathe. So how does this compare to other viruses out there? We've heard people say that this is really deadly. We've heard other people say this isn't any worse than a normal flu. So let's take a look at this graph together. What you see on the y-axis is fatality rate. That is, how easy is it for this virus to kill somebody? And on that axis, it's a logarithmic scale. So the things that are way down at the bottom are not so likely to kill you, but the ones that are clustered here at the top are much, much more likely to be fatal if you contract it. Now, what you see on the x-axis is the number of people that are infected by each sick person. If you've been listening to a lot of things that have been said about the COVID virus, that is called the R number. That is the number of people that a single infected person can go on to infect. And you can see that chickenpox, I mean, it infects lots of people. It's just incredibly contagious. So maybe for every one person who gets sick with chickenpox, 8 people get sick. Measles, it's up to 15. But you can see that chickenpox is not likely to kill you. But as you begin to move up this axis, you'll find that things like the seasonal flu, a little more likely to kill you. But, and things like MERS that we've had in the past, this was the Middle Eastern respiratory virus, the Spanish flu, the pandemic from 1918, smallpox, Ebola, those things are very, very likely to kill you. The coronavirus is in pink over here, and it's because we don't quite know how many people it kills. And we don't know that because it's easier to track the number of people who die from coronavirus than the actual number of people who have it. And without knowing what that denominator is, the number of people who actually have the disease, it is really difficult to know what that rate is. But we have some tools that we can now use. So let's talk about how we might be able to track a pandemic like this virus. So these are some of the stats that I get in the state of California because I'm part of the California Health Action Network. And pretty much every day, sometimes twice a day, I'm told what's actually going on. So as of yesterday evening, there were 6 deaths in California and a total of 335 positive cases. And again, I cannot stress this enough. We don't actually know how many total cases there are because we're unable to test a lot of people at this time. And that's hopefully going to be improving. But at the moment, that's how many deaths, and those are how many positive cases we have. Now, for those of you who are really interested, Johns Hopkins University has this amazing tracker, and it is updated not quite in real time, but almost in real time. And this is the state of COVID virus, total number of cases across the world as of yesterday, Sunday. So that is something that you can find at that link you see at the bottom of your screen. And I'll be making a lot of these links available after this talk. Now, another kind of tracker is this one called nextstrain.org. And here you can look at all these different kinds of infectious diseases, different viruses, and how they've been moving around. And this purple cluster over here over China, that was the origin of this coronavirus in Wuhan. And you will see that you can see the streams of how the virus went to all these different geographical locations. Now, as the virus moved, it mutated. It actually changed its RNA structure. And that's one of the things that can make a virus really difficult to deal with, because as they mutate their nucleic acids, their code of life, they create proteins that look a little bit different, and they make it harder for us to recognize. But we can also use this kind of information to see where it's been, how much it's changed, and how a virus found in one place might be linked to a virus found in another place because they're genetically so similar. And then we can track its spread. And you can see that here in this animation on that same site. So here we are starting in China in purple. And now you're beginning to see that the virus is diverging a little bit. It's beginning to mutate as it moves through China. And as it moves across the world, it's mutating more. Now, I do want to emphasize, it looks like there are a lot of mutational changes taking place, but compared to some other viruses, the coronavirus or COVID-19 hasn't mutated very much yet. There is some talk that there may be actually 2 strains at this time, and one of them is more lethal than the other one. So this is a good time to stop and talk about exponential growth. Here at Singularity, we're all about exponential growth. We talk about it in the context of all kinds of technologies, but it's really relevant for talking about pandemics. So this is an animation from the New York Times, and it's very beautifully done. And this is the flu virus. And let's say every one person who has the flu virus can infect about 1.3 other people. And that, again, is that R factor that I had mentioned before. After 5 cycles, there might be something like 45 people who are sick. However, something just happened. There we go. Sorry about that, everyone. With the coronavirus, you can infect 2 to 3 people for every infected person. And you can see that the growth from a single infected person after 5 cycles only can get up to 368 people sick, compared to the 45 with the flu virus. That's why this is spreading so quickly. And you can see that when you summarize these trends across the world, this early period here from January 22nd to about February 11th, they call the China period. And you can see that it has a bit of an exponential curve right there. And then it begins to spread across the world. And then suddenly in early March, it turns into a pandemic. And in fact, you truly see that in this chart. These are the total number of cases outside China. And you can see a clear exponential curve. Now, one of the problems with having exponential growth in something like a virus is, look at this, between January 22nd and February 21st, for almost an entire month, it didn't look like it was spreading very fast. And that can be incredibly deceiving. And then all of a sudden, around the end of February, it begins to take off. And you can see that this curve is changing, and then soon it shoots up. And within another couple of weeks, we have a worldwide pandemic. Now, here's a really good case illustration. This was taken actually from a broadcast of MSNBC. And you can see on the 11th, so last week, you can see how the number of cases in Italy, which we know is suffering a lot from the coronavirus right now, you can see it started at 2 cases at the end of January. And then it went to 3 cases in February. And by February 21st, it was at 17 cases, and it still seemed like it might be something manageable. But within a few days, that jumped to 219. Think about that exponential growth I was telling you about in that New York Times animation. Just another 2 days later, it jumped to 821. And then a few days after that, up to 4,000. And you can see that as of March 11th, it jumped to 12,462. Now, in the United States, where I happen to be, we had a similar sort of deceptive problem. In mid-February, we only had a few cases. And then within another week or so, we suddenly had 100 cases. And within another week, we suddenly had 1,100 cases. And if you superimpose those 2 curves, you can see that the United States is following the trajectory of Italy at this time. So we're somewhere here just as the curve is beginning to approach its knee before it shoots right up. So what do we do to contain the spread? And this is all about changing that R factor. If you have one infected person, how do you prevent them from infecting 2.3 people? How do you get that closer to 1? How do you get that closer to 0? How many of you guys have seen this video? This was actually now been made viral by by UNICEF. These are a couple of guys in Vietnam, and they're teaching everybody in this really catchy sort of tune on social media how to wash their hands for 20 seconds. And this is so incredibly important because even though it sounds trivial, of all the things you can do to stop this virus from spreading, it's to get it off your hands. So you don't touch your own face and infect yourself. So you don't touch other surfaces and you don't touch other people. Now, another way to prevent this spread, and it's something that we're all experiencing here in the United States, and in many countries in Europe and in Asia, they've already been practicing this. That's the concept of social distancing. So let's take a look at these curves. This black curve is what's going to actually happen to the spread of the virus at an exponential rate if we don't practice social distancing. That is to stop gathering in large groups, to start going to the gym, to stop going to restaurants and bars, to stop visiting our relatives in nursing homes. Now, take a look at these two lines here, the green line and this sort of burgundy line. The green line, which is centered at day 19, is, we've noticed that the curve is beginning to grow a lot and beginning to get exponential. And then we instill social distancing practices. And you can see on the green line that actually causes that curve, that exponential curve, to begin to asymptote. So we are now slowing down the rate of spread. Look at the difference one day makes. You can see that in the burgundy line. If you start social distancing only one day later, you have a huge difference in growth of the number of infected people. And again, this is because of that animation I showed you. Every one person right now has the ability to affect 2 to 3 people, and that causes things to grow out of control. Now, there was a great example of this. This is a graph that was built from data from the Spanish flu in 1918. And at that time, the United States was trying to raise money for the war. So they were holding military parades. And in Philadelphia around this time, they knew about the Spanish flu, but they decided that they were going to go ahead and they were still going to have their military parade. St. Louis, on the other hand, decided that they were not going to hold a parade. And take a look at what happened in terms of the spike in not only infected people, but in the death rate within the population in Philadelphia versus St. Louis. And I hope you notice here that this curve is huge and peaked, and the curve from St. Louis looks a lot slower and flatter. And that brings me to this concept that many of you probably heard about because the hashtag has gone viral. It's called flatten the curve. And the idea here is, here's our pandemic. It's increasing at an exponential rate. The number of cases is increasing, and the number of people who may die from this is increasing. And then at some point, it might peter out on its own. And that's if we do absolutely nothing. But we have another problem, and that's shown here on this dotted line. We only have certain capacity within our healthcare. And that was demonstrated to us by what was happening in Italy and what was happening in South Korea. There's only a certain number of medical care workers we have. There's only a certain number of ICU beds we have. There are only a certain number of ventilators we have. And if everybody gets sick and badly sick at the same time, we're going to have to start rationing healthcare. And that is not something we think about doing right now. And it's not something we're really very comfortable talking about. But in the time of a pandemic, if we can't slow the rate of spread, that's something we are going to have to consider. However, by practicing things like social distancing and hand washing, you can slowly spread that out over time so that you can get the rate at which people are getting infected to fall below that line of the healthcare system capacity. And that's what flattening the curve is all about. You may not eventually delay the total number of people who contract the virus, at least until we have a vaccine, but you will slow its spread. And you may slow its spread enough that the healthcare system can cope with it. And this brings up another thing. If you're one of those people out there who thinks that perhaps you don't need to social distance because you're young and you're unlikely to get a severe form of the disease, or you think that it's a burden on you because you don't want to decrease your social contacts, it isn't just about you. It's about the other people around you that are vulnerable. And if you are able to socially distance, you can protect all of those vulnerable people. Now, countries like China, I mean, one of the reasons that they were able to keep this under control after a while is because they implemented a lot of different things. So one of the things they did was they locked down cities. So that they basically put entire cities into quarantine. And into isolation. Quarantine means you're isolating somebody that's already been identified with the disease. Isolation just means, you know what? We don't know if you have it, but we're not going to let you get out because you're close to other people who might have it. And they built apps for this kind of thing so that you couldn't travel around China without this code and enter different provinces. That brings us to one of the most important things, and that is testing. And one thing that a lot of people don't know is, how do I even test for coronavirus? Remember, the virus is basically a ball of protein that surrounds RNA, okay? It's a nucleic acid. So one of the things that you can do in order to figure out if somebody actually has the coronavirus is you take a sample from them, for instance, from their nose or a swab from their throat, and you put it in a tube and you amplify it using something called reverse transcriptase polymerase chain reaction. The chemical details of how they do this is unimportant, but the important part is that you have to amplify the genome. And this is by far the most common way for us to detect whether or not someone has coronavirus. Another thing you can do, and this can be done after the fact, is you can look for the antibodies that a person may have made to the coronavirus. So you can take a blood sample and look for antibodies. And we do this sometimes, for instance, to figure out if somebody's already had chickenpox. They do this for me every 5 years at the hospital to see if I'm immune to hepatitis B, if I'm still immune to chickenpox. And I've gotten vaccines for hepatitis, and I was exposed to chickenpox when I was a child. And this is the way that they can confirm that I still have active immunity. What's more interesting and what's actually coming up is the possibility of using CRISPR. And for those of you who know, have heard something about CRISPR, CRISPR is considered to be this sort of molecular scissors. So you can go into DNA that has misspellings that lead to particular kinds of genetic diseases, and you can essentially do a Command F in Word and then a Command R, which is a replace. So you can find the misspelled sequence, and then you can cut and replace it with the proper spelling. In this particular case, we're going to stick to the Command F. But what we can do is, now that we know what the viral sequence is, we can identify those sequences using a particular CRISPR enzyme. And it's not the Cas9 system for those of you who are following it. It's the Cas11 or Cas12 system. So let's take a look. This is a picture of a healthcare worker in South Korea doing drive-through testing. And the reason the South Koreans were so effective at this, I mean, they have a capacity of up to 20,000 tests per day, is that people who want to get tested don't have to pay for the test, and they can just drive up in their car to essentially a healthcare worker who's in a hazmat suit. And what they do is they swab their nose, they swab their mouth, and then they can send it off for testing. Within a couple of days, the person who's come through the drive-through will find out whether or not they're corona positive. Now, the United States didn't do such a good job with this. Part of it is that we didn't recognize where we were on that exponential curve. We kept looking back in time and seeing the flat part of the curve. We didn't understand that we were coming up to that knee of the curve very quickly. But there's another reason that we were not able to respond as quickly as we should have. And that is because typically, even things like testing have to go through a regulatory cycle. They have to go to the Food and Drug Administration. And we do have something to respond a little bit more quickly called emergency use authorization, which you can see described by the FDA here. However, it wasn't enacted soon enough. Now, we have mobilized the emergency use authorization at this time, and that allows many more labs to develop tests. It allows us to get tests from other countries as long as you can demonstrate that that the test is fairly specific and sensitive. You can start testing right away. And that has led to a boom in drive-through corona testing in the United States. So this is a picture of a drive-through testing station in New Rochelle, New York. And here's another one in Colorado. And as you can see, again, you've got the healthcare workers that are sticking swabs up people's noses. However, a few days ago, Swiss drug maker Roche actually announced that they had a PCR test available, and they would be able to not only give you results quickly in 3.5 hours, but they could potentially make millions of tests available per month. So that will really change the shape of all of these curves, all the data that we have, if our testing capacity goes up. Now, going back to that CRISPR-based detection system, this is Mammoth Biosciences, founded by or co-founded by Jennifer Doudna. She was one of the people who originally defined the CRISPR-Cas9 system. Again, this is a company in which they can do viral detection using the CRISPR system. There is another company on the East Coast. This one's spun out of MIT, and this is called Sherlock Biosciences. And they're similarly using this CRISPR system in order to find viral DNA, and they are literally building a point-of-care test. You will be able to, using a paper strip, get a flow strip readout that will tell you whether or not viral RNA has been detected. So having a point-of-care test will completely change the game. So let's move on to talking about vaccines. The question is, what's actually in a vaccine? Well, there are many ways you can make a vaccine. One of them is you can inject sort of a deactivated or a non-virulent form of a virus. So basically the parts of the virus that don't get you sick. And you can inject that into a person and allow their body to mount an immune response to the thing that they would recognize if an active virus tried to invade. You can also give the person antibodies. It's not quite as effective, but you can do that passively. One of the problems is that viruses like to mutate the protein that they present. If you remember that spike protein we talked about in the beginning, we don't know yet, but that's something that might be subject to mutation. It's the reason that we don't need a constant update on our chickenpox vaccine, but we do need updates every single season for influenza because it mutates quite rapidly. And every season we'll see a new iteration of that. So one of the really interesting things coming online is that we might be able to develop vaccines now to the part of the spike protein that doesn't mutate. And if that's the case, if we can find something common that we can generate our vaccines to, we won't have to keep getting reinjected every year. A single vaccination will take care of this. So something very similar is happening now in the corona space. So at least a dozen companies are developing vaccine initiatives to deal with the coronavirus. And here are an example of some of them. And I'll just draw your attention to a couple of them. So GeoVax is creating a normal kind of vaccine, and they're in the preclinical stage right now. But there are a couple of companies. Take a look at Inovio, which is developing a DNA vaccine, and Moderna, which is developing an mRNA vaccine. Those are quite interesting because what they're doing is they're actually just putting the DNA or mRNA under the skin and allowing that to incorporate into your own cells and have you make pieces of the virus and then have you mount an immune response to it. It's actually a much faster way of building a vaccine. Moderna has been using this approach for a number of other kinds of viruses. You can see one example is Zika. The respiratory syncytial virus, for those of you who have young children, this is the thing that gets all little kids sick around their their version of a cold and flu season. And they're also doing it for things like the influenza virus. But take a look here. They're really close to actually having something that's ready to go for phase 1 testing. So finally, let's talk a little bit about other kinds of therapeutic approaches. So here's a really interesting one, and I really want to draw your attention to how recently all of this is being published. People are working at an unprecedented level to work around the world to try and find new approaches to dealing with this particular virus and this pandemic. In this case, if you look at the picture at the bottom right, what you see at the very bottom, those things that look like two sets of lollipops that are facing in two different directions, that you can imagine is one of our human cells, and that's the fatty lipid membrane that we have. And you can see that there is something green that looks like a moon with a bite taken out of it. That's called ACE2. It's something that we have on the surface of our cells. And that blue thing with all the spikes, that's the coronavirus. And the coronavirus spike protein recognizes this ACE2 as a receptor binding site. That's how it actually gets into things like our lung tissue. Now you can see that sort of purple thing sticking up on the cell membrane. That happens to be an enzyme that our cells have. It's called a protease, which it means it breaks proteins apart. And it turns out that that protease that we have is the thing that's activating the spike protein to bind to the ACE2 receptor. In other words, we're making it easier for the virus to attach to our own cells. And it turns out there is a protease inhibitor that has been approved in Japan to treat pancreatic cancer that can block the protease that allows coronavirus to attach to our own cells. So here you have a new therapeutic drug target. And this was actually discovered by a group at the German Primate Research Center. Now, here's another way we might be able to approach this. So in silico medicine is an artificial intelligence drug discovery platform. Essentially, they use computational power and machine learning algorithms in order to model the things that they want to find drugs for. And they have now turned their efforts to COVID-19. And they are even opening this up to allow other researchers to use their platform to move promising drug candidates through their pipeline. It also enables them to look at existing drugs that have already been approved and are safe for other things to see if they might work to inhibit COVID-19 or to inhibit its side effects. So basically to be used as a therapeutic. Here's something else very interesting. So I don't know how many of you about 10, 15 years ago were around to participate in a SETI project in which you would give up your computer when you weren't using it, and the processing cycles would network all the different computers to search for signals for life in the universe. So you could basically analyze the data that was coming in from their radio telescopes. There's a group at Stanford that is trying to do something very similar. They're crowdsourcing the computational power to look at how the proteins on the surface of the coronavirus might fold. And it takes an extraordinary amount of computational power to do this. But as you can see here in the quote at the bottom, for coronaviruses, the first step of infection, that protein on the surface of the virus, that's that spike protein we've been talking about. It binds to that receptor, the ACE receptor on the lung cell. But the problem is it doesn't just have one single shape. It keeps changing its shape. And because of that, we need to be able to understand how the shape changes and maybe find different therapeutics that can deal with all the different folded structures of that spike protein. And you can see here, there's a lot of different computational models that are out there now to reconstruct what coronaviruses look like and what potential drug targets there might be. So finally, that brings us to funding. How is it that we might be able to, together as a global community, finance the rapid development of both therapeutics and vaccines? So this is CEPI, the Coalition for Epidemic Preparedness Innovation. And this was founded as a public-private partnership at Davos in 2017. And they put investment money behind developing new vaccines to prevent future pandemics, and also to make the technology more democratized and available to people all over the world. But just last week, a new collaboration was announced, and the Gates Foundation, the Wellcome Trust in the UK, and MasterCard have created a $125 million therapeutics accelerator to fight COVID-19. So that's probably also going to spur a lot of innovation in this space. And that brings us to the final bit. What are our future steps? Well, one thing we can do to track things like pandemics is to use existing technology like our smartphones. Most of us have a smartphone. In fact, smartphone technology is ubiquitous throughout the world, even in places that don't have the same GDP as countries in Europe or the United States. This is an app that was developed in sort of concert between Australia and New Zealand. And what they do is they give you a bunch of screening questions, and you're asked every single day during flu season whether or not you have particular symptoms. You answer yes or no. It takes about 10 to 15 seconds, they claim, to get through all these questions. And it figures out whether or not you are likely to have the flu, and then it's able to track you and figure out whether you might be infecting other people. And that's sort of a democratized way of tracking an epidemic or making sure it doesn't get out of hand. There is another way. This is what you are seeing potentially coming out of China. It's been quite effective, but it's increased to a large degree the amount of surveillance. You can see this actually went viral. It's a video of a surveillance drone that is sort of flying over a part of China. It sees a person who is not wearing a mask, admonishes that person, and is telling her to go home. And one of the things that becomes a little bit worrisome is that this kind of surveillance and big data that's put into place in order to fight this particular epidemic, once the epidemic resolves, these particular surveillance measures may not be withdrawn. You can see a video of that here.
Like something out of science fiction, this drone equipped with a megaphone hovers before a crowd, blaring a warning. The operator, a local traffic cop.
You, the handsome man with the telephone, where's your mask? Put it on.
Hey, you on the bike, put on your mask when you're in the street. Don't endanger your fellow citizens.
Not all the drones are operated by public officials. Some of the clips have been posted by social media influencers hoping to encourage prevention.
Yes, Mom, it's the drone talking to you. You shouldn't come out without a mask. Go back inside and wash your hands. The drone can see.
I want to close with a couple of thoughts. Pandemics are not going to be rare in the future. Climate change is causing a lot of habitat destruction for animals and bringing animals and people who would normally not come into closer contact. This virus was so lethal because it was a zoonotic virus. It jumped from animals to humans, and we didn't co-evolve with them, so we weren't able to mount a more gentle immune response. Another thing in the future may be to change the way we get our meats. That's another way in which we interact with animals. Again, this was a zoonotic virus. If we were to grow meat in sterile conditions through things like cellular-based meat systems, we would essentially change the face of our interaction with animals that could potentially spread diseases to humans, especially because they're clustered together tightly in things like factory farming kinds of conditions. And finally, this is going to be solved by narrative. The way in which you teach people, say in the San Francisco Bay Area, to stop going to bars and to socially distance themselves from others is different than what you would do in Kenya or in Sierra Leone, or different in what you would do in the Wuhan province or in Paris. And therefore, you need to deploy people on the ground to tell stories that people can receive and understand. And this was done actually very successfully during the 2014 Ebola epidemic, as you can see. There were pictures painted everywhere. People told each other stories, but this all came from their local community, and it made it much, much easier for them to accept and become educated. Anyway, with that, I'm going to pass this along to my next colleague. And in fact, Dr. David Bray, who's the next speaker, is really going to go a lot into sort of the origins of pandemics and what lessons we might be able to take away from the way prior pandemics have been handled. Thank you.
Divya, thank you so much for being here with us today. I so appreciate the time, and definitely could not imagine somebody better to get us started. So with that, we are going to have to switch over to our next speaker in the interest of time. Some people are on a tight time scale today, so we're going to do our best. There are some amazing questions that I would love to get answered. So if you asked a question, we're going to figure out how to get Divya to respond to those. Please go and check Twitter at Singularity U, the letter U. And our team on social there will be following up with some of those questions, and we will find a way to get them answered. So thanks everybody for sticking around for this first session. We're going to try to make up some time. I'm going to leave here and this one, and we will drop in and see David Bray here in a second, and you will automatically get pulled into that one. Thanks.